Okay, good. So on Friday, I went to my doctor, and I've been having neck pain for like years, but it's like gotten to the point where I just can't manage it at all. And if I reach up for things, sometimes it just gets so painful, like it's just like my whole body just wants to shut down. But so I go to the doctor, and then she orders an X-ray and whatever. And it turns out I have this thing called reverse cervical lordosis, which I guess that cervical lordosis is like the curvature of your cervical spine. And mine is reverse, which means I don't have a curve. Like it's like completely straightened out. And it's caused all this inflammation, whatever. I told Dr. Morford about it because he's chiropractor, and he's who I saw first. And he's like, yeah, do these exercises, and when you're at the stoplight, push your head against your thing and whatever, and do all this. I'm like, cool, thanks, man. I don't have my physical therapy appointment until May 9th. So anything I can do to help get started, right? And today I saw Dr. Shearer. And Dr. Shearer, for those of you who don't know, he is also a chiropractor. And he was like, so basically he comes in my room, and he's like, my office, and he's like, we need to change out this chair. We'll get this other chair in here, like switches out my chairs. And he's like showing me like my posture and like how to sit. I know I have terrible posture. It's part of the problem, right? That's why this happened. Posture, computer, and this area. So it puts a lot of strain on your back after a while. So but anyways, and other stuff too. But so yeah, he's like telling me all this like advice on stuff to help with everything, and replace my chair and do these things with like a roller and all this stuff. And then he's like, okay, now we're going to adjust your back. And I said, yeah, are we? And so he literally adjusted my back like in my office. Like, that's Dr. Shearer for you though. Yeah, I mean it does. It's a little sore now, but like I've been having so many problems. That's not surprising at all. But it did. It helped. Yeah, he's a sweetheart. I don't know if I told you guys this. I think I did. I went to high school with his daughter, like literally with her. So, so like to him, I'm like his daughter basically. Okay, we'll go ahead and get this started. Remember, you can always join if you didn't get a chance to. Yes. So I don't want to go through the surgery. That's the thing, right? So it's like, other than, I mean, yeah, there's the back pain and everything like that. My sister, it's like we're all, my mom, me, my sister, we're all blessed, if you will. So she's been thinking about getting breast reduction. I just can't. I don't want to deal with the surgery and like the recovery, the scarring and the recovery. It's actually, is it not? Almost a year ago. I would say that, except like, let me put this in a perspective. I'm just being honest here. I wear a 36H. Yeah, I was a 5 at the 30. Yeah. I started at a 30G and then went up to a 34I. Yeah, so depending on the bra, yeah, it'll be I. Sometimes that I wear too, but it's like, this is like me natural. I've never had kids, nothing. Like, this is just it for me. And I'm not a fan of it, but I don't think I would want to go through healing it. I just worry. That's the part I'm terrified of. Really? Oh man, I just. I have actually heard about him, yes. Oh yeah. Did you get the surgery? Oh right, yeah. The other blessing. Yeah. Anyways, yeah, I mean, I think about it sometimes, I'm 40. I think like I'm at that point where I used to worry about if I ever had kids and stuff like that. But now I'm definitely at that age where it's like, I'm not going to have kids. And I didn't ever want to. That's not the thing. It's just, yeah, I don't know. Anyways. I really am flattered, honestly. I don't know. Yeah. I have oily skin. I think that's mostly what it is. But yeah. I mean, it sounds crazy, but it's true. Like it keeps your skin moisturized so you don't get as many wrinkles and stuff. Yeah. 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 Do any of you guys, that just reminded me, it's totally off topic, but just for a second, anybody know Dr. Smith that and he teaches like anatomy? No? Okay, never mind then. But anyway, so for the lab, now y'all having me think about that. It's saving money though. All right, anyways. It's going to be like the same exact format that we had last time, about 25 stations. It really is, I think, pretty perfectly 25 stations. 50 questions. It will be multiple choice again, 1 minute per station again, Scantron with a number 2 pencil again. All the same, nothing crazy going on there. You guys know what to expect from it. So no worries on that. All right. Any questions about like how the test is going to be given before we get into like the info stuff? Okay. Culturing basics. We're going to talk about inoculation and growth first, how you inoculate stuff, types of media. What do we most commonly use out of the list to inoculate cultures? Really difficult one. I know, right? Okay. Oh, it's okay. It's not bad right now. I got my laptop. That's all right. It'll be okay. I think sometimes about how short I am and how that's a problem too. Yeah. All right. Cool. So the loop. So in which quadrant of a streak plate, we did one of those quad streak plates, would you expect to find the most single isolated colonies? As long as I don't turn my head, it's usually not too bad. All right. Good job. We know that one. It is actually. Yeah. So culture basics, remember about aseptic technique being something that we want to avoid contamination, right? Not just pathogens, but any type of contamination into your culture or from your culture onto things. Lab safety, remember you have to wear your lab coat. Remember everybody wear your lab coat to the lab test. And closed-toed shoes. I mean, I have sandals on today, but be sure that everybody, when we do the lab exam, does have closed-toed shoes and lab coat. Remember that whenever you have broken glass, it goes in a sharps container, stuff like that. Types of media, here we're talking about slants, broths, butts, the plates, and then different ways to inoculate that media. The loop streak on a surface, or you can use the loop in a broth to swish it in there. Or you have like the needle to stab things. All right, stains. Any stain we've done up to now is fair game, technically, but I'm actually going to list on the board here in just a moment which stains you need to know, because there's some that, like I said, there's some, but there's like only 4 stains you need to know. Okay. All right. What is the first step in the gram stain procedure? All right, crystal violet. Cool. Yes, that is the primary stain. Remember, it's going to be primary is crystal violet. Then we have iodine as our mordant. We have alcohol as our decolorizer. That's on the test, by the way. And then saponin is our counter stain. What color do properly stained gram-negative bacteria stain in the gram stain procedure? Alrighty. That's going to be red. I'm not going to ever put pink on the test. So you won't have to like confuse it. What is the name of this staining procedure picture? Yeah, you guys are doing great. Do you even need me here? Because yeah, so that one's a pretty obvious one with the red being the vegetative portion and the green being the endospore itself. So which of the following bacteria will be acid fast positive? All right, so it's going to be Mycobacterium and only Mycobacterium. The other ones are false. False. Remember mycoplasma, if you see that plasma, just think of pouring plasma out. It doesn't have a shape. Why doesn't it have a shape? Because it doesn't have a cell wall. That's going to apply more, of course, to the lecture exam than this, but that's how I know what that's about. All right, after the slide has been heat fixed, let's say you heat fix your slide. Now you're doing the acid fast stain. Where do you use heat in that one? That one is a little bit of a trick. You don't use it, right? So that's going to have no heat. The version that we do in our lab called the Quignon method, 5 minutes of carbofuscin followed by a rinsing with acid alcohol and then counter staining with methylene blue. And that's it. No heat with that one. So what color is a negative acid fast stain? All right, so it's going to be blue because our counter stain is methylene blue. Carbofuscin is our first stain, our primary stain for that one. It is lipid wax soluble, so it binds with the mycolic acid wax, and it stays there. When we rinse with acid alcohol, it rinses it out of all of the cells except for those that have the wax that the carbofuscin got stuck in. So that's how we get that fuchsia stain on the positives. The negatives, though, when they got rinsed free, we have to counter stain them with methylene blue. Right, next, what is the name of the primary stain used in the capsule staining procedure? All right. Primary stain is going to be Congo red. It doesn't look red, right? So we saw the picture that was posted, right? Did you guys get a chance to look at that picture? That purple background with the kind of reddish pinkish cells with the white around them. And that white is the capsule. You're going to see the capsule stain again on this test. I feel like the sample that I picked for this particular one, because I had a couple of slides. Last one, the capsule was a little thin. This one, I feel like it's pretty obvious in this image. I think you guys will see it and know right away that that's the capsule stain. It has a background. It's the only one that has a background, right? So at least that should be a giveaway, but I'm pretty sure it's going to ask about Congo red. All righty. In which staining procedure do we use steam? Yeah, no. All right. So steam, that's endospore. Good job. You guys remember doing that with the friggin' green dye all over the place, dripping into the water and everything for 10 minutes, getting everywhere. So, all right, know the procedures. I don't care about the times that things had to sit on them. But I do want you to know the order of the different stains that you're using in each procedure and what each one is doing for the procedure. What, kind of the whole point is how it works. Be able to recognize these stains based on just seeing them in the slide. All right, moving on. Differential media, differential and selective media. What type of media is this that's pictured? Now, it's really what it looks like. And imagine if you were holding the plate, that greenish background has a shimmer to it. A metallic sheen, if you will. All righty. This is EMB. So good. So that's a good place to start. Let's say more about it here in a second. Okay, so EMB, what is the selective agent? So if you guys can remember. All righty. This is eosin methylene blue. It is the name, and that isn't meant to be any sort of like, wasn't meant to be tricky or anything. Eosin methylene blue really is the name of it. It really is a selective agent. It prevents gram positives from growing. Where does biosalts come into play? Well, we're going to get to biosalts. Don't worry. So which media is this one that is pictured? Interesting. Speak of the devil. Right. All right. That's McConkey. So that pink, and everybody puts mannitol salt, so don't beat yourself up if you did it. The pinkish background, of course, is just how that media looks, even if it wasn't inoculated on the McConkey agar. But that pink growth itself, the fact that the actual growth on it was pink is what gives it away. Because remember, in McConkey agar, if it's a pink growth, that means it is utilizing or breaking down lactose, and it turns that pink color from that lactose usage. So that's the giveaway for this one. Now, I'll tell you when I ask you a question about McConkey agar on the exam, I'm going to tell you it is McConkey. So just FYI. It's going to be like, this is McConkey agar, and then I'll ask a question about it, okay? I don't remember what the question was, or honestly, I probably would mention it, but I just don't remember. All right, is this media selective or differential? McConkey agar, is it selective or is it differential? Or is it both? Or is it all of those other things too? I don't think what else I want to put. I can't ever remember what else I write on this. Awesome. So yes, it is both selective and differential. Selective, that's the biosalts. So that's where that comes back in. That selects for enteric bacteria. Differential because we can tell if you utilize lactose or not by that pink color change. EMB has that too. EMB also has it where you can see if it aggressively uses it by that metallic sheen. What media is this? Now you guys know this one, I'm sure. But yeah, these are all of the stains that you'll need to know. By the way, just gram, acid fast, endospore, and capsule. I'm really not sure I even asked a question about acid fast on there. Yeah, mannitol salts. That's exactly right. Good job. So what is the pH indicator that we use in mannitol salts agar? Do you guys remember what it is? Kind of hard to forget, I feel like. Yeah, phenol red. So we're gonna know which one is selective versus differential, or if it's neither, or if it's both. Any of those media, remember there's a whole sheet just for those guys on Canvas. I have it in the modules area. It's like called like selective and differential summary or something. Know what the selective agents are, if it has one, and why they are selective, how that works. Understand about what makes them differential as well, what the color changes mean, if they have any pH indicators, what they are. Staphylococcus media, I am not going to ask you about it. Okay. Blood agar, it is going to be on the exam. I'm going to have a plate of blood agar. It isn't going to have anything growing on it. It's just going to be blood agar chilling. We like to remind you guys what the plate looks like because I feel like that helps people remember some of this stuff, right? But it's going to literally ask you about like one of the types of hemolysis. Like if it was this type of clearing around the growth, what type of hemolysis would be something like that. Okay. So be aware of 3 types of hemolysis. Alpha has the greening from the breakdown of the blood cells, but not complete. Beta has total clearing around the growth because we're totally breaking down the red blood cells. Gamma means you grew on the media, but you didn't cause any change to it. All right, our biochemical tests are our tube-based tests. Which one is this one? Okay. Yes, so this is citrate. The green slant is citrate. The green broth is malonate. Right. They both have the same pH indicator, which is bromothymol blue. Which of those 2 tubes is positive? Let's begin. All right, to be correct. So what is happening to the pH in tube B? All right, so in that one, in tube B, that blue color change, when we have a neutral bromothymol blue, it is green. When it is alkaline, it turns blue like that. So we have converted citrate, our sole carbon source, into basically a byproduct. And use of citrate, just to be clear, causes use of ammonia salts that are also in there, and that releases ammonia as free ammonia that is alkaline. Anyways, you guys don't need to know the chemistry of it. The good news is if you use citrate, you also cause an alkaline byproduct that causes it to turn blue. Okay, that's easy enough. Alkaline is blue, and that'll be the same for anytime you see bromothymol blue. How do we inoculate citrate? For citrate, that is a slant. So we're going to do a loop and streak it on the surface. How do we read this? Just the color change. What colors are produced? It's originally green, but it can turn blue if it is positive for citrate use. And then our pH indicator, I've said it a million times already, bromothymol blue. All right, which test is this one? Alrighty, so that one is phenol red carbohydrate fermentation. I'll tell you how I know. It's because it has that orange color to it, and it has that upside down little tube in it. You guys remember what that's called, right? Because that's going to be coming up in the question. All right, what is that upside down tube called? All right, that's the Durham tube. Good job, guys. I'm glad you know that. At least that's one thing. You don't need to study, waste your time studying that. You got that. You guys are doing really good, actually. So there could be more than one for this one. What are the two products of fermentation detected in this assay? The phenol red carbohydrate fermentation assay. What fermentation products can you tell from the results? All righty. So it's going to be gas production from the fermentation or acid production from the fermentation. So you guys are pretty good with this one. All right, this test here. What is this one? And there are probably going to be questions like that on a test, where it's literally going to be a tube, and it's going to ask, what is this test? And you're going to have to just know based on what the tube looks like. All right. Sim. How do I know this one? This is definitely one that's going to be easier. Well, maybe not necessarily easier in person. I'm not going to ask you to recognize this one in person. All of the sim tests that I'm testing you on, I'm going to tell you they're sim. And then you're just going to tell me questions about it. I will tell you that. But the reason I knew it was sim is because, number one, it looked like a butt. No, I just realized how that sounded when I said that. But yes, it did. It did look like, though, it did look like a butt. But it also looks like the tests that I know are associated with sim, which is going to be able to tell about the sulfur and the indole testing. So let's move on. What does the I stand for in sim? I just said it, of course, but still. Our indole, cool. Indole, remember, is a product of tryptophan metabolism. So when I'm asking you on the exam about the indole results, the way the question is going to be phrased is about tryptophan. So be aware of that. Me asking you two things kind of at once. How is indole detected in the sim media? What do we use? All right, addition of Kovacs reagent is correct for this one. So the reagent we add, see that little layer at the top is Kovacs, and it will turn red if it is positive. How do you detect hydrogen sulfide in sim? How do you know that it's there? The sulfur part of sim is testing for hydrogen sulfide, which is when sulfur is reduced. All right. How is motility detected in the sim test? it's gonna be growth away from the scapula, but I want to be a little bit more specific for this one. Go ahead and click forward on this. You might see a tube that looks like this. You might think you stab straight down and straight out, and you've got this stuff hanging out on the side. And you might think that that is indicating that there is motility, but that is not. This is non-motile. Why? Because that area is so clear and defined and set. If you look at what it looks like if you stab into this guy, just the same, you might see little vestiges of where there used to be a stab line, like a ghost of a stab line, but you'll also see either that there's a ton of growth on the top, so you know it's like spread all over through the media. And another thing you might see, you'll have that sort of like haziness all throughout the media. But that is more like what you would see with motility. Obviously, I gave you a tube that looked like this. I asked you, is it modal or non-modal? You could tell that that was modal. But if I give you a tube like that, you need to know that is non-modal. Okay? So I have that clear, it's just really defined looking. Even if it has little guys, it's still really defined. So I guess the better way, I say it's growth from the stab line, but what I really mean is diffusion out from the stab line. It's a better way. All right, so how you inoculate this one, that's a stab, right, with the needle. Interpreting the results and the changes. Here we're talking about for sulfur, you're looking for black precipitate. For indole, we're looking for that red color change layer in the Kovacs reagent from that golden color. And then motility for that diffusion of the growths in there. So which test is this one? Obviously, that's an uninoculated vermin. Like I'm missing something. All right, that's litmus milk. Pretty identifiable one. In litmus milk, what color is associated with an increase in the pH? All right, this is going to be blue for this one. Remember, pink is going to be acid, lavender is the original, and blue is alkaline. Now that's it for the pH stuff, but there's other stuff that can happen to it, right? Like solidifying, making a curd. Coagulation is what it calls it on there. Where it can clear up, that is digestion. But if it's white, what does that mean? All righty, so that's going to be reduction. That is correct. What does it mean by reduction? Just in chemistry, when we learned about reduction, this was chapter 10, we learned about reduction. Gaining electrons is reduction, right? So if the litmus milk was gained an electron through chemical reactions with what's going on all throughout the milk, the litmus itself, the pH indicator, becomes reduced. It gains electrons. And so it loses its color, and that leaves us with the white color of the milk. All right, so interpreting the different colors, we already went over that. With digestion, what's happening is we're breaking down all the proteins in the milk so you can see through it. How is gas detected? In that one, we're not going to see that on the test. I'm just not going to tell you that. Don't worry about it. And what is the sugar in the milk? It's not meant to be a trick question. What sugar do you guys think of when you think of milk? Lactose, exactly. Which test is this? Now, this isn't what the media itself would look like by itself. We had to add something to it, and now it looks like that. Alrighty, this is starch hydrolysis, you guys. Remember how it's going to be like a normal sort of media color, but we add something to it and it turns it that really dark color. Of course, I'm sure you can guess what's going to be the next question. All right, so first, which organism is positive in the image? All right, this is the one on the left with that big old halo around it. So what is added to detect starch hydrolysis? What did we add to that media to turn it that dark color? All right, so iodine is correct. That's what we do use. It interacts with the starch and makes it turn that dark color. The reason we see the halo is simply because there's no more starch there, so it doesn't turn dark. All right, which test is this one in the image? It is a tube with a little tube upside down in it, in case you can't see. Okay, so it's okay. Everybody's doing not their best on this question. This is nitrate reduction. Now we only have two tests, that have Durham tubes in them. That's the phenol red carbohydrate fermentation test that we already saw, and the nitrate reduction test. The nitrate reduction is the straw colored or the regular media colored one. So that's this one here. All right, what do reagents A and B detect? Have it. I may or may not have it written on board. Oh, I didn't write that part. So that nitrate test, the whole point of it is to detect if you have reduced nitrate or not. And what do we mean by that? Basically, all we have to worry about is it nitrate or is it not nitrate? That's all we really care about. But except for the fact that we need to know that if you add A and B and you turn red, that means that you are nitrite now. Does that mean we have nitrate reduction? Yes, that's a positive. But nitrite is what A and B are detecting. All right, so we had no change. The next thing is to add zinc. So you guys remember what zinc is detecting? I think is the question next. Yeah, what does zinc detect? All right, perfect. So it, yes, with zinc. Yes. So yes. So I have it written on board. So if you guys do want to come and see it afterwards, it kind of simplifies it. But we have nitrate. We're adding A and B. We turned red. That meant nitrite was there. Cool. We're done. Right. It's positive. It's nitrate reduction into nitrite. However, no color change. Next thing, be sure you add zinc. One of the questions that I tend to put on tests, I'm going to guarantee you it's on the test because I just freaking put it together. I'm going to give you a tube that we went and added A and B to, and it has no change in the color, it's still straw colored. And I'm going to ask you, is it positive or is it negative for the test? What would the answer be? You can't tell because you didn't add zinc, right? That's what the answer is going to be, FYI. So if you walk through it, you see nitrate. Is it being reduced? Add A and B, it turns red. Yes, it is, to nitrite specifically. But there was no change, so you add zinc now. So step 1, A and B, red is positive. Step 2, add zinc, red is negative. Red late means nitrate, okay? So that's how I remember it, I don't know. But red late means nitrate is still there, okay? So that's what that means, that's negative in that case. So in this case, the zinc made it turn red, not the bacteria. So that's important to remember how that worked. All right, so anyways, if it doesn't change color still, well, it didn't change color, so it's not nitrite, we ruled that out here. It's not nitrate, we ruled it out here. So it's just some other kind of nitrogen-based thing. We don't need to worry about what. So that's kind of the division now of what is what. So yeah, step 1, red is positive, nitrite is what it means. Step 2, red is negative, nitrate is what it means. That's one way to kind of break it down a little bit, but yeah, I know, I do. I told other classes, and I can tell you guys as well, if you find that you're in the test and you just cannot remember about red, what red means in the individual tests, it means positive in every single test, except for when you've had to add zinc. That's the only time, okay? So if that helps it at all, or if you get completely lost, and you're just like, God, I can't remember about the friggin' zinc rule or whatever it was, whatever that was, then just probably odds are red is positive. For the most part, that's what it means. This is just one of those weird random rules. Zinc, red is negative. All right, know what we are detecting at each step, like we were just saying. Step 1, adding A and B, we're looking for nitrite. Step 2, adding zinc, we're looking for nitrate. Red isn't always positive, like we just said. And then don't forget the gas. There is a Durham tube in there. That's just me reminding you guys that it has a Durham tube. That helps you figure out what it is. Alrighty, which test is this one? All right, this is TSIA, triple sugar iron assay. Remember how we call it triple sugar iron assay? What's that iron for? That's the chemical that recognizes the sulfur in it, that causes that black precipitate, right? All right, so which tube in that little lineup shows evidence of hydrogen sulfide production? In other words, hydrogen sulfide production or sulfur reduction. All right, good job. That's that black precipitate, right? Same as for SEM. All right, which tube or tubes shows evidence of gas production aside from those that we see with H2S? So those ones are looking aside from those. I'm gonna sneeze, I just know it. Alrighty, so it's going to be 4A and 4 because they've been lifted up out of that. All right, so how do we inoculate TSIA? You guys remember that one is a stab and a slant streak, right? How do we read it? We're going to be looking at the color changes of phenol red. Again, it could be yellow for acid production from fermentation, or it could be pink from alkaline production from using the peptone, because that's just the nitrogen source. Everything needs a nitrogen source, right? Because you got to make DNA and amino acids. What colors are produced? And what the colors mean? We kind of just said that. We just said the black precipitate. What is the pH indicator? I said that's phenol red. I do want to point out that, remember how we had talked about the slant versus the butt for the colors for this one? It was like K over K or A over A or whatever it was, right? If you were to see something that had an acid but the slant itself was pink, right? That acid but, remember, that's going to be glucose fermentation. The slant is going to be whether or not you are fermenting lactose and sucrose, the other two sugars in the triple sugar iron assay. That's all in that little table that was associated with that assignment. I'm not going to go over it too much right now, but be aware that you should probably know that for the test, because there will be a question over, there's going to be a tube, and one of the questions is going to ask if it ferments a certain sugar. And so you'll have to know based on what the color is and where it is in the tube, if it does. Okay. So that one, that's what I say when I say that TSIA comes up. There's at least 2 questions for that, and there's 2 questions for SIM, and there's 2 questions for PR fermentation. For sure, I can tell you that. Okay. Which test is pictured here? It's a little bit, the left, I would say, would be the inoculated, the one with the bacteria. The right is not. All right, so this one is urease test. It's that sort of peachy, watered down orange color, and then it'll turn that bright pink if it does have urea hydrolysis. All right, next, which test is this? All right, this one is catalase. I know our instinct is to say oxidase because oxygen production associated with it. I understand it. So I guess take this moment to be that time to remind yourself that catalase catalyzes that hydrogen peroxide into water and oxygen. We'll have oxidase next, which I think is just the answer to the question. Yeah, which test is this one? You'll know it's oxidase, but keep in mind when we're testing for oxidase, this isn't the same sort of thing, right? We're looking to see if your organism has something called cytochrome C oxidase, which is part of the electron transport chain that we see in aerobic organisms. So that's what we're looking for with this one. It's kind of a different concept. Easy. I wish, I mean, I know I didn't name it, so don't blame me. I used to confuse them all the time. And I literally did this test at my last job, and I would confuse them all the time because they'd be like, do the catalase test. I'm like, okay, let me Google that because I don't remember which one is which. All right, other tests that you want to be familiar with: malonate. Remember, that's our green broth. Phenylalanine deaminase. That's the one we added that gold drops of ferric chloride to, and it turned dark green if it was positive on the slant. MRVP, that's testing for mixed acid fermentation. Just FYI, that's the purpose for MRVP, mixed acid fermentation. Gelatin, we're looking to see if your organism actually breaks down gelatin via the enzyme gelatinase. So it becomes liquid, right? All right, real briefly reviewing over bacteria basics, and then there will be the microscope, I think. How would you classify this organism's characteristic arrangement? There's an arrow pointing at what I'm talking about. All righty. Is this a strepto because they're in chains? What is the name? It says the name and shape. Please excuse that. I mean, what is the name of the shape of these bacterial cells? So what's the shape? Alrighty. Yeah, they're rods. So they're bacilli. Good news here. Okay. You guys know, and I've told you the last time, and I'll tell you again, that the shapes of the cells you're going to see are either cocci or bacilli. I don't have anything else beyond that. So it's all you have to worry about. For arrangements of cells, if there is a question on the exam about an arrangement that involves looking at it through a microscope, it's going to be Staphylo. I don't have any other arrangement. So try not to get confused between shapes and arrangements of cells. They're not like the same thing. And remember that simple stains are a thing. One of the questions that some people miss, I don't know if there's any of you, but one of the things that was missed on the exam last time was I said about doing a simple stain with methylene blue on a gram-positive bacilli, blah, blah, blah. And people were getting confused about what color it would be. If it is a simple stain, it's going to be blue. It doesn't matter anything else about it. So it's just one color that we're putting on it. It's going to come out that color. All right, microscopes, how to use microscopes and their parts. So first of all, another question that's going to be on the test is determining the overall magnification. 100x objective lens and a 10x ocular lens. Now, I will say another thing that you want to be aware of on the test, that when you're looking at the actual slide, the objective lens is viewing it directly as its real image and creates the real image. Once it comes out through the next magnifier in the ocular lens, we're creating the virtual image. So when you look in the ocular lens, you're seeing a virtual image. Bringing it up for a reason, just say. So yeah, 10 times 100 is 1,000. It's just multiply. That'll be on the test. All right, which lens is used with oil immersion? In our lab, that's going to be the 100x. Other things for in general, parts of the microscope and what they do, the lab safety stuff, I've gone kind of over it, but all of the stains, incorrect. These stains, and maybe not focus quite as much on acid fast. I'm just saying. I just remembered that I don't think I have any questions about it. So those other three, we've really started to narrow it down. All of the tests. So all those physiological tests, I would definitely be aware of those. Those new ones that we did the second part of the semester, just they're new. I'm going to ask more about them than I am about the older stuff, right? And then identification chart and dichotomous key. If I give you guys that chart from your lab project, right? If it was yours or anybody else's, I should be able to hand you that chart and a dichotomous key based on possible results from that chart. And you can find the organism, right? There's not going to be any vagueness about it. It won't say like red, and then you have to look at the chart and say positive for. It's going to say directly what it's going to be on there. There might be a little bit of interpretation, but it's not going to be like super like ultra application time. Okay. So it's just basically testing your knowledge of your ability to take the information from that chart and go through a dichotomous key and come out with the right organism, right? There will be extra information or in there that you might not even need for the key, but you still follow. If you're gram-positive, you follow those steps. Even if it says that it was positive for indole, it doesn't show up in your steps. Hey, use the indole. Doesn't matter. You didn't need to, right? You didn't need all this stuff for your stuff to figure it out. So same concept there. Having said that, your lab reports, so I realize they are not graded. I have to grade them individually. I have to read them individually. It takes me at least 10 minutes to grade one report. Man, I even was out at the gym with my science buddies. You guys could easily see me. All of us in the science department, we go over to the gym and we'll ride like the desk bikes together. And mostly we're not doing a whole lot, but I brought my laptop and was actually working on my laptop while everybody was bullshitting. And so we'll go for like, 30 minutes or something and just like do that instead of sitting at our desks and whatever. So I even was working on you guys' stuff while I was on that. I'm still grading them. I have 2 of my courses graded completely. My Tuesday morning folks, I'm going through like in order of when I received them. My Tuesday morning folks, I have half of their class done, and then I have to do the 4 p.m. lab. That's the other half of this class. and then I get to you guys. So you guys probably won't get graded until tomorrow, but I can almost guarantee it'll be done tomorrow. But I am getting to it. It takes a while, to about 10 minutes to grade just one of them. So it's kind of a pain, but it's coming. I will say that I haven't had a single one of them. I'm about halfway, and I haven't had a single one of them be below 85 out of 100. Almost all of them, most of them are going to be like a 95 to 100. Okay. The biggest issues that I'm having are really in that sheet and filling out the sheet. It's not that I am necessarily like you put the wrong test for your organism, which is what some people were concerned about. If you get points off on it, I just don't have time to like fill out comments for reasons why I'm taking points off for every single person. It just ends up being a lot of work. So if you're interested, I can, but for the most part, in general, the reason people are losing points, if you told me that you had growth on blood auger, and that's what you put in your area, and I bring this up because almost everybody did this mistake, so don't beat yourself up if you're that person, okay? If it said growth under blood auger and you did not specify what kind of hemolysis it was, then you get a point off for that. Because that's not useful for me for identification, right? So that's kind of what I'm saying is if I were to give you a dichotomous key that depended on you telling me that, and that should have all that detailed information for all the tests. I always said that people are like sitting there like, oh shit, I'm gonna get so many points off. I'm pretty generous about this. Like if you if you're making these points off or like listing that you had results on some tests that we didn't do, it's like I'm giving like 1 point for like this fact that you got this concept wrong, and 1 point because you didn't put info on this thing, or so that's where when we're getting mostly 98s or so about it. Or so being, I'm not gonna lie, very generous on this. Yeah, that's where most of the points are going. That and the physiological tests. Of course, I'm sure nobody's surprised that would be the other area that you could see. Just because either we were getting too much description on how to do the test as opposed to how the test actually works for identification purposes. Or I had people had a good common problem was people assuming that whenever it said physiological test to test things that I meant the assigned organism. And that is not what I was talking about. I was talking, it says actually pretty clearly in the question, but I think we're just so freaked out, stressed about it. They thought that must be what I was looking for. I meant any testing of all, any kind of any differentiation, but it had to be tests that we did in the lab. So people are like describing about PCR, how this is used. And it's very nice that you're telling me this. You don't get 0 points for that. I want to be clear. You don't. I'm not going to give you 0 points for that. You get points for putting in the effort for it. It's just not going to be full points because it wasn't what was asked for. Those are the things. Any questions about that or any of the rest of this? We can see everybody's podium. Ready? Very excited. I know you don't need luck, but there it is. All right, here we go, guys. All right, good job, guys. Anyways, if you guys wanted to come take a picture of this or anything before you go, please do. I feel like I should turn the light on just for this so that. Yeah, what's up?
So like with the XDNA and like the X eukaryotes.
Right, good question. Okay, before you guys go, thank you for bringing that up. Where I have xDNA and eukaryotes and Kirby Bauer and Eliza, those will not be on the test. I know you're expected to know it. I'm not going to test you on those things. I can't fit everything on there. So you won't be tested over the DNA stuff. That includes PCR and all that. We won't, or transformation, not testing you on it. Eukaryotes, not testing you anything on them. Kirby Bauer and Eliza, I just couldn't fit them in. So yeah, those aren't on there. That's on the board, just FYI, in case you're wondering what that's about. I figured that's what you're asking. Yeah.
So another question. I just want to apologize for
Oh, please don't worry about it. You know, I don't mind. I can barely say things half the time anyway, so I'm not judging. Yeah. Yeah.
like others consider reduction should look into Dr. Shabi.
Okay.
Even though I don't like his bedside manner.
Okay.
And
Sometimes that doesn't matter as much to me. Yeah, as long as
the
they're doing
surgery
good work.
itself like was amazing.
Okay.
Like,
I might have to contact you about it if I think about it seriously because it's just like I'm terrified. I mean,
I was terrified too, but I was like, I just had to do it. Like
Yeah.
the pain, like I was getting severe migraines all the time,
Yeah.
just the tension in my head.
that's what it is.
Like-
That's exactly what I'm going through. Yeah.
As I say, a lot of people, Dr. Shabi came on. I'll show you. I don't have very many pictures because I deleted most of them from that time. Okay, so the posts. Graham. Yeah,
That's miserable.
it was super miserable. Like
Yeah, that would be painful.
it was awful. So I literally was just like, I had to get rid of them. I'm like, it was the worst.
That's the thing. That's what I hate about it the most is that like sometimes sure it's sexy, but then like other times I'm like I can't wear shirts like normal people do. Like
That was the thing.
I look vulgar in
Everything.
everything.
Like, and I will say, dating has been a huge change for me because like the type of attention I get now is completely different. It's a positive aspect. Whereas when, even though I could be fully covered, I still looked super, accentuated. I would just get the most nasty comments or whatever. And like now I don't. Now I have considered putting back in like a small and like putting in a small implant just because I lost all my upper fullness. But that's also because I've had 3 kids.
Right.
Like, he told me like if I wouldn't have had kids, then it'd probably be a lot different. I'd have like that breast tissue and stuff there.
Oh, yeah.
But that's the only thing I consider. But even if I'm scared, I'm like, no, I already got rid of all the weight. I felt good. I'm like, I don't want to put weight
It's not
in
like you
there.
were big anyway. Like if you can't.
Yeah, yeah.
Right. What? I feel like if I lost weight, I'd be